Just like skin, which is the largest organ of our body covering the external body, there is a moist lining which is coated with serous and mucous secretions that covers intestinal tract, nasal passages, and other body cavities that communicates with the external surface. This structure is known as mucous membrane. In relation to oral cavity, this membrane is known as oral mucous membrane. The oral mucosa is in many ways similar to the skin in its architecture, function, and reaction patterns. Anatomically, oral mucous membrane or oral mucosa is placed between skin and intestinal mucosa. Oral mucosa is continuous with the skin of the lip through the vermilion border, and posteriorly it is continuous with the mucosa of the pharynx. It shows properties of both epithelia. Oral cavity is a common term to denote mouth and can be divided into two parts, vestibular cavity and oral cavity proper. Vestibular cavity is bounded by lips and cheek on the outer aspect, while teeth and gingiva is going to line the inner aspect. Oral cavity proper lies interior to the palate or inner aspect of teeth, bounded superiorly by palate, inferiorly by tongue, and posterolaterally by facial pillars and tonsils. Color of oral mucosa depends upon four factors: degree of keratinization, thickness of epithelium, proximity to the underlying blood vessel, and fourth factor is the presence of melanin pigment in the epithelium. Color of a healthy oral mucosa is pink. Lateral parts of palate gives a yellow color because of the presence of fat in the connective tissue. There are some clinical considerations. An anemic individual present with a pale colored oral mucosa, especially labial mucosa, whereas in some conditions we can see dilatation of blood vessels presenting an erythematous appearance, giving a clue about the pathology. Consistency and thickness varies in different regions. Consistency can be firm or soft depending on the location. For example, palate is firm, buccal mucosa is soft. Thickness ranges from 0.3 to 6.7 millimeter in the oral mucous membrane. Thinnest areas are floor of mouth and ventral portion of tongue, and thickest region is maxillary tuberosity. Oral mucosa has many important functions. The oral mucosa protects the deeper tissues and organs from the mechanical stresses or forces of mastication and external environment. It prevents entry of bacteria and their toxins by forming a physical barrier and antibody secretion. The commensal organisms of the oral cavity becomes pathogenic if the host defense is compromised. So one of the main functions is protection and defense. Next is sensation. Receptors in the oral cavity respond to touch, pain, temperature, and proprioception, and also enable reflexes like swallowing, gagging, retching, and salivation. The taste buds on the tongue help to perceive taste sensation. Next function is sensation. Oral mucosa contains minor salivary glands in its submucosal part. Saliva secreted by major and minor salivary glands is discharged onto the oral cavity through the ducts, and it results in the moistness of the oral mucosa, which is going to keep the oral environment healthy. The oral mucosa might not have an important role of thermal regulation in humans, but in some animals like dogs, some of the body heat is dissipated through panting. Next function is absorption. Oral mucosa absorbs fluid electrolytes, and drugs by various mechanisms. Drugs kept in direct contact with the oral mucosa are more readily absorbed than those dissolved in saliva. The absorption potential of oral mucosa is greater than the skin. Excretion. Some metabolites are excreted through oral mucous membrane. The excretion is another function of oral mucosa. Color and amount of visibility of the gingiva and color of the lip mucosa or labial mucosa enhance facial aesthetics. So these are the functions of oral mucosa: protection, defense, sensory, secretion or lubrication, thermal regulation, absorption, excretion, and finally role in aesthetics. Based on function, oral mucosa is divided into three types: masticatory, lining, and specialized mucosa. Masticatory mucosa, covering 25% of oral mucosa, is bound to bone and does not stretch. It bears forces generated on it when food is chewed. Gingiva and hard palate is lined by masticatory mucosa. Lining mucosa, on the other hand, is not equally exposed to such forces. It covers the muscles related to oral cavity and is adaptable to movements produced by muscles of mastication. It covers 60% of all oral mucosa. That is, all the surfaces of the mouth except for the dorsum of the tongue and the masticatory mucosa. Rest 15% is lined by specialized or sensory mucosa. It is so called because it bears the taste buds which have a sensory function. Based on the type of keratinization, oral mucosa can be keratinized mucosa or non-keratinized mucosa. With this, we are going to see what keratinized mucosa is. These are the components of a keratinized mucosa. There is an epithelium, connective tissue, and submucosa. Epithelium, a stratified squamous epithelium, consists of squamous or flattened epithelial cells arranged in layers upon a basal membrane. Only one layer is in contact with the basement membrane. The other layers adhere to one another to maintain structural integrity. Lamina propria represents a connective tissue of variable thickness that supports the epithelium, which is continuous with submucosa. But in areas of mucoperiosteal attachment, there is a direct attachment of lamina propria with the underlying periosteum of bone. Let's discuss oral epithelium in detail. Keratinized stratified squamous epithelium can be orthokeratinized or parakeratinized depending on the presence of nuclei in the topmost layer. In case of keratinized epithelium in oral mucous membrane, it is usually parakeratinized. Epithelium connective tissue interface is marked by corrugations which gives a serpentine shape. The portion of epithelium projecting towards connective tissue is called as rete ridges or rete peaks, since it resembles specks. At the same time, portion of connective tissue that projects towards epithelium is connective tissue papilla, which carries blood vessels and nourishes epithelium. Like other epithelia, the oral epithelium maintains its structural integrity by a process of continuous renewal in which cells produced by mitotic divisions in the deepest layers mature and undergo terminal differentiation as they migrate passively towards the surface to replace those that are shed. The terminologies associated with this continuous renewal are maturation, differentiation, desquamation, and turnover rate. Maturation is a process of cell migration from basal layer to the surface. Differentiation is the biochemical and morphological changes of cell during its migration. That means its movement from basal cell layer to the keratinized squama. Desquamation is a shedding off of the superficial cell, and finally, overall time taken for cell migration and desquamation is the turnover rate. Turnover rate is 52 to 75 days for skin, 41 to 57 days for gingiva, 25 days for cheek, and 14 days for intestinal mucosa. Cell proliferation in the epithelium is checked by growth inhibitors by maturing cells. This is a schematic picture of a keratinized stratified squamous epithelium. Talking about histology of keratinized epithelium, 90% of cells are keratinocytes, which is also present in the non-keratinized epithelium. What makes keratinocytes unique is the presence of keratin tonofilaments, which forms the intermediate filament contributing to the cytoskeletal structure, and also the presence of desmosomes, which are the intercellular bridges. The intermediate filament protein present is cytokeratin. Functions of cytokeratins are they form the cytoskeleton of epithelial cells and maintain the shape of the cells. Cytokeratin act as a stress-bearing structure within the epithelial cells, and they also help in distributing the forces over a wide area and provide mechanical linkage. There are 20 types of cytokeratins: type 1 or basic cytokeratins and type 2 or acidic cytokeratins. They always occur in pairs of type 1 with type 2. In the absence of its pair, they are unstable and are susceptible to degradation by proteases. Cytokeratins are tissue and layer specific. For example, suprabasal layers of masticatory mucosa express cytokeratin 1 and 10, whereas cytokeratin 4 and 13 are expressed by suprabasal layers or suprabasal cells of lining mucosa. Based on the morphological appearance, keratinized epithelium consists of 4 layers: stratum basale, stratum spinosum, stratum granulosum, and stratum corneal. This is a schematic picture showing 4 layers of a keratinized epithelium. First layer is stratum basale. It is represented by single layer of cuboidal or columnar cells that has the ability to undergo mitosis. Cell is characterized by centrally placed hyperchromatic nuclei with basophilic cytoplasm. Active protein synthesis for basal lamina and for intermediate filament is noted. This layer consists of 2 cell populations. One is serrated cell population packed with tonofilaments which aids in attachment. Second group is non-serrated cells. They are the dividing cells. Non-serrated cells can be further subdivided into slow dividing cells, which are going to preserve genetic material, and large amplifying cells. Basal and parabasal layers together constitute stratum germinativum. Stratum spinosum is a second layer which is made up of polyhedral cells with spiny or prickly appearance. That's why the name stratum spinosum or prickle cell layer. These cells are larger than the basal cells and are joined by prominent intercellular bridges. Now, what is the reason for spiny appearance? During tissue processing, cell shrinks and make intercellular bridges more prominent, giving away a spiny appearance. It resembles a plant variant cocklebur. Involucrin, which is a precursor protein for cornified layer, is first seen in stratum spinosum. This layer Stratum granulosum contains flatter and wider cells. These cells are larger than spinous cells. Cells contain basophilic keratohyalin granules, favoring dehydration and keratinization of cells. Nuclei will start showing degeneration and pycnosis. Tonofilaments are denser in quantity. Involucrin, present in upper half of the stratum spinosum, contributes to thickening of the inner unit or inner aspect of cell membrane, forming a conified cell envelope. Arguably, the most famous border wall, the Great Wall of China, is one of the world's largest building construction projects. It is basically built to serve as a barrier. Similarly, we have a permeability barrier between granular and corneum layer, which is formed with the help of Ordland bodies. Ordland body or keratinosome are also called as membrane-coated granules. They are seen in the upper spinous and granular cell layer. These are small, membrane-bound structures of 250 nanometer size and originates from the Golgi bodies. In keratinized epithelium, the granules are elongated and contain a series of parallel lamellae. In non-keratinized epithelium, they appear to be circular. In stratum corneum, cells are larger and flatter than granular cells and are eosinophilic in nature. They present the degenerated nuclei and other cell organelles. The cells are composed of densely packed filament coated by basic protein of keratohyalin granule filaggrin. Disulfide bonds closely pack the tonofilaments and provide chemical and mechanical resistance to this layer. There are two types of maturation of keratinized cells, orthokeratinization and parakeratinization. What we just discussed is orthokeratinization. In parakeratinized epithelium, the superficial cell layer retains shrunken or pycnotic nucleus. This is a picture representing parakeratinization. Here we can appreciate pycnotic nuclei. That's all about keratinized epithelium. Keratinized epithelium is always supported by lamina propria or connective tissue and is continuous by submucosa, which later attaches to the underlying structure. These are my references. Thank you.